Today in this video we are going to discuss one of the important experiments called as Hersey and Chase experiment, which was conducted in 1952. Now this experiment is a confirmatory experiment. Why? Because after Griffith experiment, as you know that Griffith experiment is known as transforming principle, where they were unable to conclude the factor that cause transformation. So later on, as we know that McCarthy, MacLeod and Avery have performed the experiment, and they come up to a conclusion that it was DNA that is responsible for the transformation. Now this experiment will confirm that DNA is the genetic material. Let's see what Hersey and Chase have performed the experiment. Now, what they have done was they used T2 phases, which is also known as bacteriophage. T2 phase, bacteriophage. What is T2 phase or bacteriophage? These are those viruses that infect the bacteria, and they can infect even plants and animals. Let's try to see the structure of this bacteriophage. It has a polygonal sheet like this, and they have a spiral protein. There will be an end plate, okay? This is said to be end plate, and you'll find tail, like this type of structure. So this is said to be tail plate or end plate, okay? Now this polygonal sheet, you can say this is capsid protein, and inside there is a DNA as a genetic material. At the middle there is a perforation. Perforation at the middle. Now this is the structure of your bacteriophage. Now what did they have done was they took these T2 phases and grow in a media where the media has sulfur 35 and phosphorus 32. Now these are radioactive elements. So they grew these T2 phase. They grow these T2 phase in a media or in a medium, okay, where there is a sulfur 35 and phosphorus 32. Now remember, this sulfur 35 is not a part of DNA, it is a part of protein. So it has to be incorporated in the amino acids. As you know that proteins are made up of amino acids, right? So this sulfur 35 was incorporated in a amino acid, that is cysteine and methionine. Okay, cysteine and methionine. Whereas phosphorus 32 was incorporated in DNA. So as they were grown in this media, it means that now we got two types of T2 phases, okay, where just say that this is two bacteriophage, where in this phase it was incorporated with sulfur 35, and in this bacteriophage it was incorporated with phosphorus 32. Okay. Now, what did they do was, second step, they allowed these phage to grow in a host. Since virus they cannot multiply without a host, so they use the host as E. coli. Just say that this is E. coli and this is our phage attacking on the host cell. Just say that this the process as infection. Okay, infection. Now, after infecting, what did they do was they go for blending. Blending means mixing. Okay. After blending, they go for centrifugation. Centrifugation. What is centrifugation? Okay. Actually, this sample was in a test tube. So they took in a test tube and allowed to grow, infect, and they put in a machine called as centrifuge. Okay. Now in this centrifuge, they use centrifugal force so that they will rotate in a very high speed, known as RPM. Okay, RPM, revolution per minute. Okay. Now, after that, what they found? Okay. So this is a test tube. Just say that this is E. coli. Okay. This is E. coli cells. So this the upper part is said to be supernatant, and the lower part is said to be palate. Now in case 1, just say that this is case 1 or case A or B. Okay. In the case A, they found that the supernatant, okay, they found that the radioactivity was in a supernatant. In this case, the radioactivity was in a supernatant. In this case, the radioactivity was in a E. coli. Okay, it means that the radioactivity was not found in the supernatant. The radioactivity was not found in a supernatant. Okay, but found in a lower part, that is the E. coli. So from this experiment, they come up to a conclusion that if protein was a genetic material that had entered into the host cell, they will find the radioactivity in a lower part. Okay, but it was not so. The protein code we have labeled. Okay, we have labeled the sulfur 35 as a radioactive substance in a protein code, in this code. Okay, and the phosphorus 32 was labeled in a DNA. Fine. So when radioactivity was observed after the experiment, okay, they found that the radioactivity was in a E. coli cells. It means that it was a DNA that had entered into the host cells. So this experiment confirms, okay, Hersey and Chase experiment confirmed that DNA is a genetic material in all the living organisms. Now, in the next topic, we are going to discuss one of the important experiments which prove that DNA replication is semi-conservative. In previous class, we have already discussed what is DNA replication. It means that copying of DNA to DNA. Now, what does semi mean? Semi means partly. Okay, partly. You can say partly or half. Okay, and conservative, it it is talking about the all things. When I say all things, it can be all strand. Okay, since we are talking about the DNA. Now, it means that DNA replication is conservative, means just say that this is 3' to 5' and this is 5', 3'. Okay, complementary strand or anti-parallel. So when they go for replication, you know that this is 3' to 5', 5' to 3'. So this is the parental strand or the all strand. Now you get the another newly synthesized strand, 5', 3', 3', 5', right? So see, one is old strand and one is new strand. Semi, partly half, 50% from the parental strand and 50% is newly strand. Okay, so this is said to be semi-conservative mode of DNA replication. Now, to prove this, Meselsen and Stahl had performed the experiment to prove the semi-conservative mode of DNA replication. Now, what did they have done was they took heavy nitrogen. Okay, they took heavy nitrogen. So that heavy nitrogen was N15. Now there was a source. Okay, they took a source. The source was ammonium chloride. So they took this ammonium chloride, which contains heavy nitrogen, and grow in a media. They have added in a media, and the E. coli were allowed to grow for many generations. So these bacteria started multiplying for many generations, and it contained their DNA will consist of heavy nitrogen. So this is 5' to 3', 3' to 5'. Okay. Now let me assume that this is N15 and 15. Okay. So both the strands have a heavy nitrogen N15. Now what did they do was they took this strand, all strand, and allowed to grow in a normal nitrogen. So normal nitrogen was also provided in the form of ammonium chloride. But see, normal nitrogen means N14. Okay, so for 20 minutes, okay. So what happened was, in the first generation, okay, so this is our parental strand, N15 and N15. Okay. So this just say that this is our newly synthesized strand. But here in this media we have nitrogen 14. So this newly synthesized will have nitrogen 14. Just say that this is generation 1. Okay. Now in the next generation, the same thing will happen. They will undergo replication, and the strand will be separated. Just say that this is nitrogen 15. Okay. This is nitrogen 14. Another strand, nitrogen 15. Another strand, nitrogen 14. So for this strand also they have added in normal nitrogen, that is ammonium chloride. So let's see the newly synthesized strand. So you can see that all the newly synthesized strand will have nitrogen 14, that is normal nitrogen. Now these DNA fragments were separated. Okay, they separated these DNA fragments with the help of density gradient. I'll write here density gradient. and this density gradient is cesium chloride. They observed that with the help of density gradient method, they observed that for this observation, I am writing here, they observe at the end of the test tube there is a heavy nitrogen. For this generation, they found that they found a band at the middle. Okay, so I'll just write as hybrid. Okay, and for this observation, they found two bands. So one band will be showing lighter one. Okay, lighter means containing both. This is the one N14 and 14, and this is the one N14 and 14, and these two are hybrid. Hybrid. This is lighter because it is having normal nitrogen. Now this experiment, you can see that the old strand which contained nitrogen 15, okay, as a parental strand. See in generation 1, one is newly synthesized and the other is the parental strand. Now if you allow to grow in the next generation again, in generation 2, again you found that there is a new strand, okay, and there is a parental strand. It means that partly half means 50 is to 50. Okay, so this experiment, okay, proves that DNA replication is semi-conservative. There was another scientist, okay, who is known as Stahl, who had performed the same experiment in DNA and chromosome. Okay, Stahl in DNA and chromosome. In the root tip of Vicia faba. So it's a name of the plant, okay? But they didn't use nitrogen 15, but they used thymine with heavy hydrogens. So this experiment, when we say semi-conservative, it means that it is partly half and partly from the all parental strand. In the next class, we are going to discuss about packaging of DNA.